All right, class. So thank you very much for being patient. I'm a little bit under the weather today. But hey, this is partly why we study human biology, because as human beings, we have human bodies and we get sick. And in order to help create new treatments and new cures for diseases, we have to understand the human body and understand how it works. So, Dr. Todd will be filling in today. I know you will all treat him with just as much respect as you treat me, so thank you very much for your patience. So today's topic is going to be on DNA extraction and DNA fingerprinting. DNA, or deoxyribonucleic acid, is the genetic material found in living organisms. So this is where we store our blueprint for how to build a human body. DNA is located inside the cell. Most of your DNA is found in the nucleus, as we talked about before. However, a part of your DNA is found in the mitochondria. So it's a very small portion. And by looking at mitochondrial DNA, this is very important for ancestry purposes, because this can tell you your maternal lineage going all the way back. So by looking at the mitochondrial DNA, scientists are able to figure out what your ancestral family tree looked like thousands and thousands of years ago. So the cell membrane and the nuclear membranes are made out of protein and lipid. I prefer to say lipid and protein because most of it's made out of this lipid bilayer, which is basically fat. Um, Proteins can do a couple different things in their membrane. So they can span the membrane, making like these doorways between the inside and outside of the cell. They can also float on the membrane like rafts, doing various functions. So the structure of DNA, DNA is what's used to store the information inside of your cell. This is made out of a series of nucleotides. You remember we learned about A, T, C, and G last week. So A, or adenine, T, or thymine, C, or cytosine, G, or guanine. So DNA is a computer. It's what stores the information inside of your cell, as I said. And it's also used to process a lot of information. So while a computer made by human beings uses ones and zeros, DNA uses 4 bases. So it uses the A, T, C, and G system. So instead of being digital, it's more like a quaternary system. As you can see here, DNA is double-stranded. We have one strand here and one strand over here. And you have it where A always pairs with T, T always pairs with A, C always pairs with G, and G always pairs with C. And these are connected by hydrogen bonds, as you can see right here. And this ends up arranging itself into a double helix. So you can see here how this almost goes up like a spiral ladder or like a spiral staircase. And you have the major groove here is the wider part and the minor groove right here. So the two strands, the two spaces between them are not equal in their length. And that's why we have the major and the minor groove here. And as you can see here, we have a book that says The Fat Cat Sat on the Mat. And that's similar to how the bases in DNA record information. So you have a series where it says GCG, and so 3 nucleotides will make 1 codon, and then CCC will make another codon, etc. And this system ends up being what codes for the blueprint for a protein. Now, not all of your DNA codes for proteins, and that's okay, we'll get into that here soon, but when you learn about the genetic code, what that means is that for every 3 letters or 3 nucleotides in a protein coding sequence, those 3 nucleotides will come together to form a codon or a word, which ends up being translated later on into an amino acid. And amino acids are what makes up the chains of proteins. So as you see on the slide, the order or sequence of these bases determines the information available for building and maintaining an organism, like the way in which letters of the alphabet appear in a certain order to form words and sentences. So the information here has meaning. It's not just a random series of A's, G's, C's, and T's. This is a blueprint which codes for a particular protein, which gets translated later on. All right, so today we're going to be using split peas to extract their DNA. So one of the first things you're going to do is let the split peas sit in cold water for 15 to 20 minutes. Hopefully we started doing that before the lecture. And then we're going to add a cubic centimeter of salt. This helps DNA to precipitate by neutralizing the negative charges for every 300 milliliters of cold split pea solution, and blend on high for 15 seconds. So if you have 300 milliliters of solution, you use 1 cubic centimeter of salt. If you have 600 milliliters, then you would use 2 cubic centimeters of salt. You're going to pour the suspension through a strainer or through a cheesecloth. So this big solution of split pea, pour it through this strainer, and you'll get a little bit of green liquid at the end of that. And then what you're going to do is add a 15 milliliter amount of liquid detergent. This is going to help break up the whatever's left over from the cell membranes. And this is what detergent does, is it takes all those lipids that are attracted to one another, breaks them apart, and helps to mix them in with the water. And this is why you use detergent when you're washing dishes. Allow mixture to rest in ice bath for 10 minutes, so you're going to let it get cold, which is very important. And then you're going to pour this into a test tube. It's going to be about a third full, tilt at about a 45-degree angle, and you want to slowly pour the alcohol over the top. We should be using isopropyl today, and this bilayer is going to help to pull the DNA out of solution, and it's going to look like clear snot. And you're going to use a toothpick or like a coffee stir, and you're going to just swirl that around gently, not to mix things, just gently, and you're going to gradually wind that DNA around the toothpick or the coffee stir. All right. There's also a virtual lab available. If you do the physical lab to extract the DNA, you should be fine. You should be able to answer the questions with that. Now, if Dr. Todd has you click on this link, great. If not, I'm going to consider it to be optional. So click on this link if you want to use the extraction, but the directions on the previous slide should be sufficient for what we're doing today. All right, so as we can see on your slide, genes are small segments of DNA located on chromosomes. A gene is a set of segments of nucleic acid that contain the information necessary to produce a functional RNA product in a controlled manner. RNA in turn makes a gene product, most often a protein. So, that was a mouthful. What does all that mean? Well, you often hear about genes in the media or genes in pop culture. Basically, along your chromosome, very small portions of it are what we call genes. So these are the parts that actually code for proteins. And these only make up a very small percentage of your DNA. Now, the traditional understanding was that one gene would code for one protein, and proteins do most of the jobs in the cell. But now, as we've understood genetics more, we understand that one gene can code for multiple proteins. So we expected there to be around 100,000 genes in the human body, in the human genome at first, but now we know it's much, much less. So your genes are basically the blueprints for proteins. However, they can also code for things like ribozymes or functional RNA molecules. All right, so the Human Genome Project. This was completed in 2003, and the Human Genome Project was a 13-year project coordinated by the U.S. Department of Energy and the National Institutes of Health. Now, this was a very interesting time in the history of science because you had this publicly funded side of the Human Genome Project, and then you had private sector side led by Craig Venter, and the two were kind of in an arms race to see who could map the human genome first. So, very interesting time in the history of science, definitely something to look into. So the project goals were to identify all of the approximately 20,000 to 25,000 genes in human DNA. At the time, we thought there was going to be close to 100,000, but like I said, it turns out that one gene can code for more than one protein, and there's a whole bunch of reasons for that that are beyond the scope of what we're doing here. And this is also to determine the sequence of, it was actually over 3 billion chemical bases that make up human DNA. We wanted to take this massive amount of information, more than 3 billion bases, and store this information in databases for future use so that we could better understand how the human genome works and learn ways to make cures for diseases, make treatments for diseases, learn better ways to make medicine later on. And we also wanted to improve tools for data analysis. We wanted to transfer this technology to the private sector and address ethical and legal issues that might arise as a result of the project. Now, in 2011, when I was finishing up my associate's degree, the professor challenged us. He said, we spent all this money on the Human Genome Project. What has it given us? And I'm not sure if he was against the Human Genome Project or if he was just challenging us to think. However, the question's valid. What was the result of all of this? And part of the answer that I would give in retrospect would be that if you plant an apple tree, don't expect to get fruit tomorrow. Sometimes it takes a while to profit from your investment. And since the early 2010s, there's been a lot of improvement in terms of things like personalized medicine, in terms of ancestry technology, and I would also argue that the social aspects, the fact that we know that everybody on Earth is very, very close in our DNA, that should really give us some implications to start thinking more about things in terms of ethics, thinking about things in terms of realizing that that other human being is also a human being. So that's part of what I would say to that. All right, so genes are a series of nucleotides that code for proteins. So as I mentioned, the genes are the portions of your DNA that codes for proteins. Most of your DNA does not code for proteins. Most of your DNA is made out of things like regulatory sequences or structural sequences, and there's portions of your DNA that people debate, does this have any function at all? So that's an ongoing debate. However, my opinion on things is to say, hey, don't say this piece of DNA has no function just because we don't know what it is yet. So over 99% of human genes are the same from person to person. A very small percent of genes, 0.1%, code for the differences between people. One of my very first science jobs was to look through data from the 1,000 Genomes Project. And this was a project where they'd collected DNA from over 1,000 different people and from all over the world. And it was my job to scroll through all those differences, and I was writing a computer program to analyze them. And it really hit home for me that whether you have somebody from Northern Europe, Central Africa, or East Asia, everybody in the world has almost the exact same DNA. If you were to have an alien beam down and look at human DNA from any two people anywhere in the world, he would say, hey, we're basically all clones of one another. So a lot of times people will say, well, I don't like this person from, this other country or from this other continent, and they'll say, well, this person's a different race from me. But in reality, what we've really learned from studying DNA is that there is only one race, the human race. We are all basically identical to one another in terms of our DNA. One little tribe of chimpanzees has more genetic diversity within their tribe than the whole of the human race put together. So there's really, if you just don't like somebody, okay, fine. I can't convince you necessarily to like somebody. But this idea that we're different races or that there's something different between us, it's really not true. Just because somebody looks a little different from you, that's not reason to hate them. We are almost identical to one another, no matter where in the world you come from. And humans and chimps differ by only 1.2%. So you'll often hear humans and chimps are 99% or 98% the same. It kind of depends on how you run the algorithms, depends on how you calculate the similarity or how you calculate the differences. Um, but I want you to take a minute to really process the implications of this and process the implications of this for society. The humans and chimps, that our DNA is so close to one another, and how does that scientific fact impact your view of the world? So not all DNA is used for coding genes, of course. There's repetitive sequences of nucleotides that are located between gene sequences. So in this case, we have ACG, ACG, ACG. And this does not code for a gene. You'll see this over and over again. And this is what we call a variable number tandem repeat. So what that means is variable number tandem repeats, they're not genes, they don't code for anything, but the length of those variable number tandem repeats will vary from person to person. And the number of times these, just said that. So the length of the sequence is unique for each individual, and this length is genetically determined. So why do we care about that? Why is this important? I'm glad you asked. So VNTRs help to identify individuals in DNA fingerprinting. So the steps in DNA fingerprinting would involve PCR, step one. So there's polymerase chain reaction, which is basically like a Xerox machine taking a piece of DNA and copying it over and over again exponentially, and we'll see that here in a minute. You can also split the DNA into fragments using restriction enzymes. So these restriction enzymes will come in and cut the DNA at a very specific sequence. And so that's 4 to 8 nucleotides long, and that gives you a bunch of fragments of DNA that can be used to see your DNA fingerprint. So these restriction fragments, which are different lengths because of the NTRs, they result in fragments. So the polymerase chain reaction. So we have multiple PCR cycles, start with one piece of DNA, you get 2 copies, 4 copies, 8 copies. So this grows exponentially. And you see exponential growth a lot in biology, whether it's rabbits breeding in the wild or bacteria breeding in your test tube, or in this case, making multiple copies of a piece of DNA. So this is a molecular biology technique for enzymatically replicating DNA without using a living organism. The technique allows a small amount of DNA to be amplified exponentially. So it means it keeps going up and up and up, and the rate that it goes up keeps getting bigger. Because it is an in vitro technique, it can be performed without requirements on the form of DNA, and it can be extensively modified to perform a wide variety of manipulations. So PCR is used for a lot of different things. It's used in COVID testing labs, it's used in clinical labs for other things, it's used in research. Anywhere you use DNA, there's probably a PCR instrument somewhere. So the fragments separated by gel electrophoresis. There's So what we do is we make a little agarose gel, and we put the fragments of DNA, we put the samples of DNA in there. They're stained, and then this is passed through a gel. And the gel kind of looks like a Jello cup, like your mom might have given you when you were a kid. And we pass this through the gel using an electrical current. So ion buffer solution is poured over gel to help conduct electricity. So the ions are used to help conduct the electricity because water just by itself won't conduct electricity. You need to have the ions in there. So you need to put salts in there or something along those lines. The samples are loaded into wells. The current is turned on with positive charge anode applied to one end and a negative charge or a cathode applied to the other. The fragments, of course, move according to size. So as the DNA is moving through the gel, it's kind of like fish swimming through a series of nets. The tiny fish will swim very fast through the net, whereas the big fish will get caught more easily. And with DNA being these long strings, as they're going through this net structure, which is what agarose gel looks like if you were to look at it under a super duper powerful microscope, they get caught in the ropes of the net. and gradually move as they go. So that's why smaller fragments are able to move faster than other ones. And the end result ends up being like this barcode type pattern. So very convenient that this is what DNA does. Very convenient that this is what it looks like on an agarose gel after we run it. All right, so the number of alleles. The number of times that a sequence is repeated varies from individual and between the maternal and paternal loci of an individual. So between your mother and your father, the DNA from your mother and your father, you're going to get different numbers of these tandem repeats. So there is a 1 to 100 chance of having a matching allele to another person for a particular allele. So this particular variable number tandem repeat, you might have the same one as somebody else, but it's a 1 in 100 chance. But as the number of alleles that are compared increases, the probability of having a match becomes smaller. So if you're just looking at one allele, the chances of sharing the same one with another person be about 1 in 100. But if you start looking at 10 alleles, then the odds of having the exact same DNA fingerprint increases exponentially, or rather it decreases exponentially in this case. So the gel electrophoresis. So we can use a virtual lab to run a gel, and you click on the following link. I'm going to make this one optional. If you would like to click on it, great. It's a lot of good stuff on there to learn. You'll learn how a gel electrophoresis works. That said, we're already trying to do 2 things in lab today, so I'm going to make this one optional for you. If you do it, great. Please let me know. Otherwise, it's optional. So you can go through the entire lab taking notes as you go. Your lab handout might have a brief outline, but it's up to you to fill in the details. And we're actually not going to turn in the answer sheet today. I want you to hold on to the answer sheet, and you're going to turn it in through my courses using I created a virtual quiz in my courses, and it's multiple choice, 4 possible answers for each multiple choice, and you get 10 chances to get it right. So 10 chances to get a multiple choice of 4 questions correctly. So I do want you to fill out that sheet, and then after you fill out that sheet, you're going to use it to do the My Courses assignment. So we're trying to do a lot all in 1 week, and again, thank you everybody very much for your patience. Please don't stress about these things. We're going to make it all work, and it's my goal to make sure everybody gets an A in this class. Now, of course, that's on you to get the A. Your grade is your responsibility. But I'm not going to deduct points because a stupid computer system's not working correctly. All right, so Clue, the game of Who Did It. I have not played Clue in my lifetime, so the other professors ran through this with me and explained to me how the game of Clue works. We have the rules right here. Work together to kind of figure out the rules. Instead of using weapons, we're using a piece of forensic evidence. So you might say it was Mr. Smith in the dining room with the dirty tissue. So instead of saying Mr. Smith in the dining room with the candle stand, it's Mr. Smith in the dining room with the dirty tissue. So there's also going to be printouts on the clue cards of your DNA fingerprint. So obviously you're adults, don't worry so much about winning or losing. Just work together to kind of play the game, figure out how it works, and use the game to write down notes, maybe take pictures so that you can fill out those 8 questions that are at the end of lab, because those are what you're going to use for your online My Courses multiple choice. All right, so this is a fun optional reading. It is not required. It is not part of your grade. So Professor Richard Buggs wrote this blog post saying, how similar are human and chimpanzee genomes? So I mentioned before the idea that human beings are 98 to 99% the same, and I said this is a fact, asked you, hey, how does this fact impact society? But just because your professor calls something a fact does not necessarily make it true. So I wanted to use this as an opportunity for critical thinking, because we hear this 98 to 99% number all over the place. It's in the media, it's everywhere. And I think even most people who are geneticists don't necessarily understand where this 98 to 99% figure comes from. So basically, what happens with this 98 to 99% figure is we're looking at the parts of the human and chimp genome that are very similar, but this is excluding the parts that are very different. So it's looking at the parts that are homologous, so the parts that are very similar between humans and chimps, but the parts that are not homologous, that are totally different and we don't know why, those parts were excluded from these estimates. So Professor Richard Buggs, he's an evolutionary biologist and a molecular ecologist, he explains that he thinks that when everything is really said and done with a lot of these analyses, humans and chimps will probably turn out to be more closely to 85% the same. So I wanted to put that out there to help people understand that we have this, sometimes we have memes going around in science, sometimes we have these pieces of information that get spread around, and sometimes there's a little more to the story that people might not necessarily be aware of. So that's something to keep in mind when you're learning about science, you're learning about scientific ideas. So if you would like to read that article, it's right here. And definitely let me know what you think if you read the article. All right, so your to-do list for this week. You're going to have the pDNA isolation. You're going to have the forensic clue. Have fun with this. It's not that big of a deal if we can't figure out the game exactly, as long as you're learning from it. And the same with the pDNA isolation. I'm not going to give you a bad grade if you mess up isolating DNA from peas. It's not that big of a deal. I want you to all have fun, and even if everybody said, hey, we can't get this stupid snot to work in this pea soup, fine. That's okay. That I'm not going to take away points for that. But try to use these things and use these things to learn. So you also have gel electrophoresis using an online module. This is optional. And your actual points for this week, after you do these things in lab, it's going to come from the My Courses Online assignment, and that is what you're going to submit for this week. So thank you so much, class. Thank you for giving all your respect to Dr. Todd, and I look forward to seeing you next week.